Nucleic acids. Nucleic acids are naturally occurring chemical compounds that is capable of being broken down to yield phosphoric acid, sugars, and a mixture of organic bases. They are the main information-carrying molecules of the cell and by directing the process of protein synthesis. Sometimes it feels like DNA get all the credit. Of course, yes, the structure of DNA is amazing and has a beautiful double helix structure and a twisted ladder. It has its own emoji. DNA stores the genetic information. DNA is a genetic material that transfers the genetic information from one organism to their offspring. It is located in nucleus and mitochondria. 99% of bases are same. The order of bases determines the individuality. The information in DNA is stored as a code. In 1952, Alfred Hersey and Martha Chase confirmed that DNA is a genetic material. James Watson and Francis Crick found that DNA is made up of two polynucleotides. DNA is a long-chain polymer of nucleotides. It is a double helix with two strands which gives ladder-like shape with base pairs. The base is attached to the sugar by N-glycosidic bond. Nucleoside is converted into nucleotide by attachment of a phosphate group. The linkage between the nucleotides in a polynucleotide is a phosphodiester bond. It consists of deoxyribose, which is a pentose sugar, phosphate group, organic bases like purines and pyrimidines.
However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first before DNA in RNA world hypothesis. A nucleic acid that carries the genetic message from DNA to ribosomes and is involved in the process of protein synthesis is referred to as RNA or ribose nucleic acid. Ribonucleic acid helps in the synthesis of proteins in our body. It is a polymer of ribonucleotides linked together by phosphodiester linkage. RNA is similar to DNA but contains ribose in place of deoxyribose. RNA exists in several different single-stranded structures, most of which are directly or indirectly involved in protein synthesis or its regulation. It also acts as a genetic material in some viruses. The three main components of RNA are phosphate group, ribose sugar, and nitrogenous base. RNA acts as a very important biomolecule. It facilitates the translation of DNA into proteins. It functions as an adapter molecule in protein synthesis. It serves as a messenger between the DNA and ribosomes. It is a carrier of genetic information in all living cells. It promotes the ribosomes to choose the right amino acid required in building new proteins. RNA does not form an analogous double helical structure, but RNA forms a heteromeric double helix.
Transferred ribonucleic acid tRNA is a type of RNA molecule that helps decoding a messenger RNA, that is mRNA, sequence into a protein. tRNAs function at specific sites in the ribosome during translation, which is a process that synthesizes a protein from an mRNA molecule. tRNA molecule has distinctive folded structure with three hairpin loops that form the shape of a three-leaved clover. One of these hairpin loops contain a sequence called the anticodon, which can recognize and decode an mRNA codon. Each tRNA has its corresponding amino acid attached to its end, When tRNA recognizes and binds to its corresponding codon in the ribosome, the tRNA transfers the appropriate amino acid to the end of the growing amino acid chain.
Messenger RNA is a single-stranded RNA molecule that is complementary to one of the DNA strand of a gene. It carries a portion of DNA code to other parts of the cell for processing. It created during transcription. During protein synthesis, an organelle called a ribosome moves along mRNA. Read its base sequence. It uses the genetic code to translate its 3 base triplet or codon into its corresponding amino acid. All mRNAs are read in the 5' to 3' direction, and polypeptide chains are synthesized from amino acid to carboxylic terminals. Each group of 3 bases in mRNA constitute a codon, and each codon specifies a particular amino acid. mRNA sequence used as a template to assemble the chain of amino acid that form a protein.
Characteristics of ribosomal RNA are important in evolution, taxonomy, and medicine. Ribosomal ribonucleic acid is a type of non-coding RNA which is primary component of ribosomes essential to all the cells. It serves critical role in forming the catalytic sites of translation of mRNA. Both prokaryote and eukaryote ribosomes can be broken down into 2 subunits, one larger and one smaller subunit. Ribosomal RNA is a type of non-coding RNA which is the primary component of ribosomes. Ribosomal RNA is transcribed from rDNA and then bound to ribosomal proteins to form the ribosomal subunits. rRNA is a ribosome which carries out the protein synthesis in ribosomes.